Tuffs University. Dr. van Deventer received his bachelor's in chemical engineering at Stanford University in 2004 and his PhD in chemical engineering from David Terrell's lab at Caltech in 2011. After that, he did a postdoc in Dane Wittrup's lab at MIT, where he was Ludwig Foundation for Cancer Research postdoctoral fellow and an NIH Ruth Kirstein National Research Service Award fellow. After his postdoc, Dr. van Deventer accepted an assistant professor position at Tufts in 2015, where he has received funding from at least NIH that I could find, an R21 and an R35. Broadly speaking, his research uses a combination of synthetic biology and chemical biology to establish new strategies for treating cancer and other diseases. More specifically, his laboratory is currently focused on disrupting enzymatic and signaling processes within the tumor microenvironment by leveraging the chemical versatility of yeast display to discover new kinds of inhibitors with novel chemical compounds, which of course he does by pioneering new genetic code expansion platforms in yeast. Collectively, his work has resulted in at least 32 peer-reviewed publications. Dr. van Deventer, it's a pleasure to have you with us today. Looking forward to your talk. Take it away.
Thank you very much for the opportunity to share some of our group's work. So what I'm going to focus on today is I'm going to focus on applications of genetic code expansion in yeast, and in particular, what we're really motivated by in part of the research group is we're motivated by the fact that if you look at antibodies, they're amazing as therapeutics, and they're also highly engineerable. However, on the other hand, there's a lot of functionalities in antibodies that are not accessible because of the limitations of the conventional genetic code. If we look at small molecules, we sort of see some of the opposite sets of features where we can see some really interesting covalent functionalities that can be introduced within small molecules, as well as targeting enzyme active sites in ways that are really difficult with antibodies. But the specificity of small molecules sometimes leaves something to be desired. So for these reasons and others, we're really interested in figuring out all of the different ways that we can try and combine the best of these both worlds. So how can we make hybrid structures that can retain the best features of proteins and small molecules, but also gain something beyond what either one of those things can do alone? And so that's really driving the vision of the research group. We're interested in therapeutic development, but also in establishing diagnostics and even basic research tools where the existing molecules that we have access to are just not able to do the things that we want them to do. So to nobody's surprise, we're using genetic code expansion to achieve part of this vision. And so I don't really think I need to say too much about adding amino acids to the genetic code. But one of the ways in which our group is a little bit different from others in the community is our focus is almost entirely on yeast and doing genetic code expansion in yeast. And the primary reason why we have chosen to focus on yeast-based genetic code expansion is so that we can access really powerful protein engineering technologies. So I'm showing here a schematic representation of yeast display. And yeast display is this really powerful protein engineering tool that allows us to do high throughput screening, but it also allows us to do high throughput characterization. We can evaluate binding functions, and as I'll show you today, we can also evaluate properties that we add into proteins during the course of genetic code expansion. So things like covalent target engagement, conjugation reactions, and the properties of not just proteins themselves, but also of the hybrids that we display on the yeast surface. So before I get into sort of the specific examples I'll talk about today, part of my group is focused on how is it that we can push the limits of genetic code manipulation in yeast. So we make very careful measurements to understand how the systems work. We make attempts to enhance the genetic code manipulation machinery in yeast in various ways, including genome-wide screening and other approaches. And I'm happy to talk about that offline. But for the purposes of today's talk, we'll be focusing on how it is we can start exploiting the chemistries that we can genetically encode in yeast-displayed proteins for various applications. So the way that we've been trying to set up these problems is as follows here. So we know that we can display proteins containing non-canonical amino acids on the yeast surface. The one set of questions that we need to ask ourselves when we start doing this is okay, like, how do we want to try and enhance the functions of proteins? Do we want covalent target engagement? Do we want to present pharmacophores? Are there other chemical features that we need proteins to have? So that drives what chemical elements that we attempt to add to yeast-displayed proteins. And that's a really important set of technological questions. But then on top of that, we also have to ask ourselves, what should we do once we can introduce different chemistry into proteins? Should we take existing antibodies and other proteins and try to systematically study their properties and enhance the properties of existing proteins? Or should we be a little bit bolder? And should we actually do high throughput discovery where we're actually starting with a large collection of chemically expanded proteins and then we screen in that format? So for the purposes of today's talk, we're going to focus on sort of the top set of questions, is how can we take existing proteins and try and expand their functionalities by adding additional chemistries into them. But hopefully in the future, I'll be able to share work related to our library discovery efforts, which are ongoing. So the first story that we're going to talk about for a little bit is a story where we've attempted to engineer covalent antibodies. And again, we've done this in our yeast display platform so that we can do it very efficiently. And really to discuss why it is that this might be something that we should do is really to think about what is it that we want binding proteins to do. So there are cases where it's difficult to engineer binding proteins that last for long enough or that engage with their targets for long enough. Covalent target engagement is one strategy for trying to make these binding proteins act for longer. But there's also a number of other cases where a covalent engagement event could be very useful. For example, diagnostics depend on non-covalent interactions typically, but dissociation in diagnostics leads to loss of sensitivity. So wouldn't it be great if we had access to covalent binding reagents that we could put into diagnostics? The same thing is true with protein-protein interactions, where some of these are very, very transient events. And if we could make covalent forms of these protein-protein interactions, that might actually be really useful for understanding what these interactions are doing. So for all of these reasons and more, we wanted to see what would happen if we started encoding these different covalent functionalities within proteins in yeast display format and seeing, well, what can we actually discover if we can expand the genetic code with these particular amino acids. So the particular system that we decided to use to begin to study this was a really well-developed system that was established by collaborators of ours. Charles Shoemaker at Tufts University discovered a series of single-domain antibodies that then have been characterized in a high level of structural detail by Rongsheng Jin at UC Irvine. And all of this work has also been done in collaboration with a group at NYU School of Medicine, the Chinko Lab at the NYU School of Medicine. So all of this structural data existed when we started collaborating with these groups, but this particular system is a really challenging system here. And the reason why I say that is because the protein that is being targeted by these single domain antibodies is a really incredible protein. This is the light chain protease of botulinum neurotoxin. And this protease, when it gets inside of neurons, it causes neuronal paralysis and eventually death if the infection is bad enough. And this protease can last for months inside of neurons. So if you think about it from a treatment perspective, this is really, really challenging. where any sort of dissociation event of an antibody or another therapeutic from the protease, that could cause reactivation of the protease. And so one strategy for trying to overcome this extremely long-lived protease that, if it becomes active, can do really terrible things, is to introduce covalent functionality into antibodies. And so as an initial proof of concept that we could convert existing single domain antibodies into covalent reagents, we established a yeast-based approach to do this using sets of proteins that our collaborators made available to us. So basically, we were able to display a series of proteins on yeast and then evaluate whether or not these proteins were engaging covalently using some very efficient assays that a former postdoc from the group, Rafa Alcala Tarano, developed. So let me walk you through some of the data that we generated for this project, focusing on the efficiency with which we can use this combination of yeast display and non-canonical amino acids to find these proteins with expanded properties that you couldn't get otherwise. So we first started by creating a pretty extensive series of substituted mutants. So these are 4 different single domain antibodies, and we picked 5 to 6 non-canonical amino acid substitution sets for each of these proteins. And then Rafa very meticulously verified that substitutions at all of these positions result in retention of binding activity for everything that we looked at. So that was really nice to be able to confirm that. It was also relatively straightforward to confirm that because we could do these binding assays in yeast display format. The next thing that I'll spend a little bit more time highlighting is the fact that once we had these proteins and we had verified that we were able to bind to our targets, we could start studying the cross-linking properties of these proteins. So this amino acid here, azidophenylalanine, it's a very popular genetic code expansion amino acid. But for those of you who might not have seen it before, this is a UV activatable cross-linking amino acid, where if you shine UV light on it, it will be converted into a highly reactive form that will covalently bond with anything that's nearby. So we took this amino acid and systematically substituted it into all of the different single domain antibody variants that we had generated. And then on the yeast surface, we ran through all of these proteins and established a workflow for first incubating everything with the target, shining UV light on the mixtures, and then very stringently washing away all of the non-covalently bound target, and then running everything out in a flow cytometer, asking the question, can we detect the target after getting rid of all of the non-covalent interactions? And so elevated signal in this assay is an indication that we're observing covalent binding events. And so this detection scheme here is one where all of the controls are present and showing these low levels of target detection in this bar graph. But then when we start looking at some of the substituted variants, such as this series in red here, you can see that many of these variants, when we run them through this assay, end up showing very high levels of target detection in comparison to the controls. So this is a really strong indication that what we've done is we've identified variants here that are covalently engaging with this protease target. So this was a really encouraging series of results here. On the other hand, you could argue that, well, azidophenylalanine is one of the most reactive amino acids that you could possibly introduce into a protein. So what happens if we tune down the reactivity of a cross-linking amino acid by quite a lot in this system? Can we still discover cross-linkable forms of binding proteins? And so to study that, we turned to this proximity-induced cross-linking amino acid here. So this amino acid is known as a weak electrophile. Historically, it's thought to react with cysteine residues primarily, but we and others have at least some evidence that it can react with other nucleophilic amino acids as well. And so we decided, well, let's do the same experiment that we just did with the photo cross-linking substitutions, but let's now do proximity-based cross-linking substitutions. And so after Rafa promoted cross-linking for a pretty extensive period of time for the purposes of assay development, we then washed away all of the non-covalently bound proteins as best as we could, ran everything in the flow cytometer, and the data that we got out was very different from the data that we got for the photo cross-linking. And what you can see here is you can see that there are these three substitutions or these three variants that show elevated levels of cross-linking, and everything else is at or below the control levels of target detection here. So this is a really exciting set of results here because it shows that by tuning down the cross-linking reactivity, that reduces the number of cross-linking solutions that we identified in our yeast display survey here. And so this result in itself is very encouraging. For the sake of time, I am not going to tell you about all of the follow-up solution studies that we did, except to say that we verified some of the key cross-linking events that we observed first on the yeast surface in solution. We also did extensive series of specificity assays in solution, and really that the cross-linking events that we identified in the cross-linking variants, it looks like they're pretty specific for the target of interest. And so we think that all of this really points to some promising approaches for how to discover covalent antibodies in high throughput. So this is one way that we have implemented our technology for adding chemical functionality into binding proteins. The second way that I'm going to focus on for at least a couple of minutes to give you a flavor for it is to use small molecule pharmacophores, where we're presenting those within binding protein structures. And so this work was very recently published. Literally like a day or two ago is when the final publication came out. So let me give you a flavor for this. So I'm showing here some pharmacophores that are capable of targeting carbonic anhydrases. The good news about these pharmacophores is that there are examples of quite potent pharmacophores that are known to inhibit the enzymatic activity of carbonic anhydrases. The bad news is that in humans, there are 12 enzymatically isoforms of carbonic anhydrases, and we really don't have good inhibitors that can distinguish between these different isoforms of carbonic anhydrases. So it'd be really great if we could figure out how to take these pharmacophores and make them into more specific inhibitors of these proteins. So we're not the only ones who have been thinking about this. In fact, the laboratory of Benjamin Hackle at the University of Minnesota has thought about this challenge quite a lot, and we've been fortunate enough to be collaborating with his lab on this. It turns out that in a really nice recent study, Ben's lab did a whole series of high throughput discovery involving this pharmacophore here, conjugated to proteins known as fibronectins. These are antibody alternatives that also allow for very tight binding interactions to occur. And in the Hackle lab, what they did is they very cleverly used some thiolmaleimide chemistry in order to present this particular warhead within their proteins of interest. The result out of their screening was that they found some isoform selective inhibitors, and this was really exciting work. Now, when we started collaborating with the Hackle lab, we said, well, this is great. How far chemically can we change around these inhibitors and still end up with interesting inhibitors? And so this study that we performed in collaboration with their lab is one in which we started with some of their inhibitors, and then we swapped out the thiols for clickable non-canonical amino acids. And we combined those with this series of pharmacophores that I showed you a couple of slides earlier. And you note that all of these pharmacophores have these clickable functionalities on them. So this enabled us to produce 48 new hybrid structures that we could then evaluate for their inhibitory properties and their binding properties and other characteristics. And so very briefly, this was enabled again by the fact that we can do this work on the yeast surface, and we can do it very efficiently because we're never making purified proteins in order to generate these constructs here. So we can make all 48 of these hybrids at once, and that's exactly what a recent PhD graduate, Manjie Huang, did. She made everything all at once. And then in doing that, really expanded the chemical space of these hybrids beyond what had been explored previously. So very briefly, we took some of these clones, and if we kept the same pharmacophore that we started with, or that the Hackle lab had started with, we observed lots of binding activity against the two carbonic anhydrase isoforms that we had seen, that had been seen before. So this was encouraging, and it shows us that there's actually quite a bit of chemical tolerance in these hybrids to begin with. But what's even more exciting is when we explored other warheads, we discovered that substitutions of this pharmacophore into these hybrid structures still resulted in some really nice active hybrid structures in our initial survey. So this was also really promising news, and really told us that, hey, like there really is a lot of chemical territory that we can start to access using this yeast display strategy that we've established here. So, so being skeptical, we said, well, like just how well performing are these particular hybrids, to look at these things efficiently, we made a lot of measurements on the yeast surface in order to determine that as far as we can tell, a lot of these new candidates that we isolated are nanomolar affinity hybrids. Some single-digit nanomolar, some double-digit nanomolar, and some triple-digit nanomolar. So not all of them are super high affinity necessarily, but they all show a good amount of activity. And then what was even more encouraging is that when we took this work into solution, and we made a few hybrids in solution, we observed pretty potent enzyme inhibitor properties for the hybrids that we evaluated in solution. So this is a good portion of this story that was recently published, but for the sake of time, I think that's where we should wrap it up for today. But I'd encourage you to take a look at this manuscript in order for further discussion of this work here. So in summary, this hybrid space that we've been able to efficiently explore in yeast display format is a really exciting one that demonstrates the power of manipulating these chemical elements. We think that in combination with higher throughput screens, this is really going to give us access to all sorts of new inhibitors and other types of proteins that can modulate biological processes in new ways. And so with that, I'll just conclude with these exciting opportunities that we are starting to open up with our technology platform. And I'll stop there and take a couple of questions if there's time for it.
Great. Thanks, Jim. That was really, really fantastic work. Very, very exciting to hear. We do have time for a few questions. So if we'll open up the floor to anybody who has questions. Looks like we have one from Ryan.
Jim, really nice work. Excited by your proximity-induced cross-linking and your small molecule pharmacophore work. It's really, I just wanted to sort of clarify, because of your use of yeast display, my understanding would be that you don't really need highly efficient reactions to do the decoration of new molecules, because even in yeast display, you're going to have many thousands of proteins displayed, and even if only, say, 10 or 20% of those reactions move to completion, that would allow you really effective evaluation of different sites or different chemistries really quickly. So you don't have to have good reactions. The reactions don't have to run for long, if I'm correct. Am I reading that correctly?
Yes, yeah. I mean, so those are really important features of our yeast display format. So absolutely, if we're only seeing reactions that are happening at 10% of the available displayed proteins, that's fine. We can easily detect that using yeast display. So that's true for chemical reactions, where you're trying to do a conjugation. But I think more importantly, as you've noted, it's true for cases where we're trying to figure out how to grab on to a target of interest.
Yeah.
So even if we've got a very efficient proximity-induced cross-linking reaction, or we've got a very weak binding affinity for a target, we can detect that, and then we can pull that out, and we can think about, okay, great, like we've got something to, we've got a starting point here. Where do we take it from there? And that's where all of the yeast display, antibody affinity maturation strategies and things like that can be used to really, push these things even further.
No, it's a wonderful, wonderful discovery platform. I totally agree. And then, the optimization later on with different amino acids or different chemistry or, whatnot is really easy to probe and validate on more single molecule sort of scenarios. Beautiful work. Thank you.
Thank you.
Cool. Thanks, Ryan. Any other questions?
I just actually, I haven't been paying a lot of attention to yeast work lately. And my follow-up question would be, in the context, so the feeling in the field for genetic code expansion has always been that, yeast genetic code expansion is lower yielding than other systems. And right now you've done quite a bit of optimization in these areas. If you were looking at sort of yields of protein toe to toe, say with E. coli systems, how would you compare them right now, and what are the spaces for improvement?
Yeah, so that's a really good question. Okay, so if you start, so there's really two aspects of this that are important. One aspect is how efficient are the stop codon suppression systems in yeast in comparison to the stop codon suppression systems in E. coli. And so we, in our lab, we've characterized systems that are pretty efficient. You can observe 50% stop codon read-through in non-canonical amino acid incorporation using some of the characterization systems we've established in the lab. And so from that perspective, that's not really limiting in terms of being able to do things in yeast. So the efficiency of stop codon suppression is not too bad. It's not at the level of a genomically recoded organism like you have available in E. coli, but it's still quite good for a lot of applications. Now, in terms of producing proteins in yeast versus E. coli, that's where you run into a different set of challenges. So in laboratory settings, before you bring non-canonical amino acids into the picture, E. coli systems, you can get, several hundred milligrams per liter of protein expression. For yeast systems, and again, this is before we involve non-canonical amino acids, the systems that we've done secretion with in our hands, maybe if we're doing really well, we can get something like 5 milligrams of protein out of those secretion systems. And so that there is where you might take a hit in terms of protein yields. And then if you add on top of that inefficient genetic code manipulation systems, then that hits you further in terms of how much protein you're producing. So at this point, and I guess the other thing that's really important to remember about Saccharomyces cerevisiae, which is where we do most of our work. Actually, it's where we do all of our work right now. Saccharomyces cerevisiae is not really set up to be a great protein secretion platform for a lot of applications. There are some examples of industrial strains that do quite well and can give you gram per liter yields of proteins, but But for the laboratory strains we've been using, it's not really set up to be great at protein production. If you had your choice between producing a non-canonical amino acid containing protein for structural studies in E. coli versus yeast, I would, almost always choose E. coli, just because the yields you can get in E. coli are probably going to be a lot higher. That being said, I think that the discovery platform that we have in yeast is something that does really well for itself. Are there ways to get better protein expression in yeast? Yeah, definitely there are, but how much do you need it if you're gonna be mostly doing discovery, and then you want to do single molecule characterizations using, whatever the best system is to produce your material?
Oh no, I totally agree with you. And you're not looking for massive yields for this particular project. I was just trying to understand sort of the logistical differences between the two situations, since it's something that comes up a lot. And I appreciate the thorough answer. It's a really great conversation about the pros and cons of the two situations. And there are definitely many situations that really do require yeast. And so I'm excited about this area growing. Thank you.
Thank you.
Great. Thanks, Ryan. We have 1 minute for anybody else who has a quick question. I'll just ask a real quick one. Well, I guess we're out of time, but just real quick, are there thoughts or plans for basically combining the two parts of your talk, basically taking this sort of these inhibitors and tethering them to antibodies for the botulin protease that might sort of combine together as a potentially more extra effective therapeutic?
Yeah, so can we start adding multiple chemical elements into the same proteins to try and push their performance even further? Is that your question?
Yeah, it's effectively the broader scope picture. Yeah.
Yeah, so we recently reported that we can use two distinct non-canonical amino acids in proteins that we can prepare in yeast. So that gives us some more opportunities to add multiple chemical elements into proteins. But as other groups have shown, the thiolmalamide chemistry is something that works quite well. That's something that we could consider combining with non-canonical amino acids as well. So all of these things are things that we're thinking about.
Great, cool. All right, so we are at time. Thank you to both of our speakers, Jim and Natalie.